All right, thank you for that introduction. So turkeys, like all poultry, are long day seasonal breeders. So they're photoperiodic, and thus that means that light is what initiates activation of the reproductive axis, so exposure to a long day photoperiod. And in birds, in comparison to mammals who receive the signal via the eye, in birds, the signal is actually perceived in the deep brain. So light must first penetrate the skull and brain tissues to reach deep brain photoreceptors in the hypothalamus, which initiate activation of the reproductive axis. And in female poultry, that means the initiation of lay. So while photoperiod, which is the light duration as measured in hours, can serve as the trigger to the onset of reproduction, there's actually been some more recent interest into how other parameters, so namely intensity and spectrum, so light intensity being the available light energy as measured in watts per meter squared, and spectrum being color or wavelength range as measured in nanometers, and how those can impact potentially reproductive performance in birds. And that's mainly due to the fact that light must first penetrate the skull and brain tissues, and intensity means it measures the amount of energy that can actually reach those photoreceptors, and spectrum has to do with the actual ability, so tissue penetrability of light to reach those photoreceptors as well. So in practice, the turkey industry recommends using very high light intensities for their breeder flocks, so 100 lux, which is up to 10 or 20-fold what is used in commercial layers. And for turkeys, there is not so much empirical data showing that this might actually improve reproductive performance, but there is quite a lot of evidence that shows that it may compromise welfare across poultry species, but including turkeys as well. On the other hand, the use of red light has been shown consistently across poultry species to improve reproductive performance in layers, broiler breeders, geese, and even some wild bird species. However, this had yet been reported in turkeys. So we hypothesized that we can improve reproductive performance through a more pronounced stimulation of the reproductive axis by stimulating a larger population of photoreceptors via increasing the red light output of our light source. And that's because red light is more easily able to penetrate tissues and reach the photoreceptors, and through increasing light intensity, since we're increasing the instantaneous available photic energy. Our objectives of this trial were to determine the effect of a 60% red spectrum, so that's 60% red, 20% green, and 20% blue light, or cool white LED at high or low intensity on female turkey reproductive capacity. So that's through egg production and estradiol profiles, and then explore any consequences of light treatment on egg quality. So in this trial, we placed 402 grandparent female line hybrid turkey hens at 20 weeks of age into 4 rooms. So 2 were illuminated with our red spectrum light and 2 with the cool white fluorescent. And we maintained all these, both of these rooms, all these pens under low intensity light for the remainder of the rearing phase. So that was from 20 to 29 weeks of age. And these actual light treatments corresponded to 0.1 watts per meter squared in terms of energy, and they were harmonized between the 2 color treatments. So white and red had the same amount of energy, although you can see that this differed in lux, which is why we didn't use lux to measure energy per se. And if we look at the actual spectral distribution of these 2 light sources, we can see that under our red spectrum light, the majority of the energy peaks in the red spectrum, versus under a cool white fluorescent, we have this spike in the blue and relatively even distribution across the rest of the spectrum. And so this is what our experimental setup looked like. Now, during the lay phase, we photostimulated these birds at 30 weeks of age with a 13.5-hour photo period. And this is when we actually introduced our second light treatment. So that was intensity, and that was assigned by Penn for a split plot design. During lay, we collected eggs 12 times daily and evicted the birds from the nest at each egg collection and removed floor eggs. So that's what would be done in a commercial setting. That's what we also did in the research setting. Now, if we look at this, our high intensity white treatment corresponded to 100 lux. So that's the commercial recommended intensity. And that was threefold in energy to the low intensity, but harmonized between color treatments. We collected blood samples of focal birds weekly around the time of maturation, so 29 to 36 weeks of age, and then bi-weekly until the end of the experiment, until 60 weeks of age. We took bi-weekly body weights of all individuals, and then we measured egg quality parameters, so weight, breaking strength, and shell thickness of all eggs for 3 consecutive days every 4 weeks throughout lay. All data were analyzed in SAS with a 2 or 3-way ANOVA and Tukey's post hoc test with repeated measures, and differences were considered significant at p less than 0.05. So moving straight on to the results, we saw no significant effect of light treatment, so color nor intensity on egg production performance. So you can see, to orient you, our high intensity treatment is in the broken lines and low solid lines, red in red. And then if we look at the cumulative egg production, we saw a 3 egg per hen house increase under high intensity red light. And I would like to emphasize that this is not statistically significant. However, it is quite in line with what we see in the field in other species and what we have previously seen in the field with the turkeys. So it warrants further investigation. Now, in terms of the plasma estradiol profiles, we saw this is pretty interesting because this is the first time this has been characterized in turkeys, so estradiol profiles throughout lay. And we were interested to see that there is a second peak around 48 weeks of age in all groups. And overall, this was also interesting because, contrary to our hypothesis, we saw higher estradiol levels under white light, which is a bit difficult to explain at this point because egg production didn't differ. But hopefully we'll be able to understand this better once we do some molecular analyses. Now, in terms of body weight, we saw during the growth phase slightly higher body weights under red light. However, this difference dissipated at the onset of lay. So during lay, under low intensity light, we had significantly higher body weights. And then this is the uniformity as measured with the coefficient of variation. So this is body weight uniformity. And we saw no difference during the growth phase, but during the lay phase, we saw improved uniformity under the low intensity light. Now, last, I'll show you the egg quality parameters. So as expected, we saw an increase in egg weight with age. However, we also saw higher egg weights under low intensity light, and that's likely due to the fact that these birds had higher body weight as well. So in conclusion, we saw no impact of light treatment on production, although we did see that 2.5% increase under high intensity red light, which we're hoping to look into further. We also saw that increased body weight under low intensity light with improved uniformity, and that was potentially due to reduced activity. However, those behavioral analyses are in the works right now. We also saw an increased egg weight under low intensity light, which could have a potential impact on hatchability and poult quality. And we'd love to investigate these impacts in a commercial setting to see how this might impact hatchability and poult quality. And so with that, I'd like to acknowledge my advisor, Dr. Gregor Beta-Carrots, and colleagues, the undergraduate students who helped me with this, and the Canadian Poultry Research Station staff, as well as our funding partners. And I would love to take any questions if anyone has any. Thanks.
Right, thank you very much, Clara, for that great presentation. In terms of behavior, because here we are still in a behavior and welfare session, and I know that you haven't finished analyzing those behavior, but like kind of from running the experiment, are you anticipated potential effect on, for example, aggression or from your treatments?
So anecdotally speaking, I didn't see much difference. I would expect that we'll see higher aggression under higher intensity light, since that's what's been previously reported in the literature. But in terms of feather damage and other sort of anecdotal observations, I didn't see any differences.
All right, thank you. And I see Val has a question. Could you go again over the lighting paradigm that you used during the rearing?
During the rearing phase. So during the rearing phase,
prior, maybe I think the question maybe relates to prior to placement of your birds.
Oh, prior to placement. Oh, yeah, that's a great question. So these birds were reared in a commercial facility prior to placing them at the research station. So from hatch to 19 weeks of age, they would have been reared under the commercial, according to the commercial guidelines in a commercial facility. Good question. Thanks. So really, they've been exposed to 2 to 3 different light treatments.
Yes, so because I believe the question was related to could that rearing part could have affecting the response to your treatment?
Yes, yeah, it's possible, but they were all reared under the same conditions. So yes, it's possible, but in terms of what we saw in this experiment, at least that was controlled for.